All right. Hello, everybody. Happy Friday. Okay, so usually I go around and just check names so that I remember, but I don't want to be late. I had Nigel in the back. Check you off if you're here. I'm gonna go around and say your name really quick. T-I-M, Tam. Hallie. Lisa, Sweet. Yes. Sarah, Jason, Chris, not taking the test. Ash, Carol, Kevin, Taylor.
That's next week.
Oh.
Friday the 16th.
Okay, yes, that is Priyanshi.
I can go to Kumar.
You want me to call you Kumar because I keep forgetting your first name?
Gosh,
Gosh, I will try to remember your first name. That's Kumar for now. No.
Ian.
Trina? Or Katrina? It's Katrina. Ah, it's Katrina, but we can call you Trina. And Kevin. Medora. Beautiful name. Stella. All right. Dan. Okay, great. Okay, you guys. Let's keep going. I realized something last Wednesday. I recorded the lecture, but without sharing my screen. So you only hear my voice. So now I'm recording with sharing my screen. Okay. So just recap, remember every participation is a +2. Love to hear your voice, love to talk to you. And it's okay if the answer is not correct. You're here to learn. All right. Why do we analyze food from a consumer perspective? And hello, just state your name for Nigel. Braden. Okay. Anybody want to give the first question a try?
Safety,
Quality, safety, yes. So consumers care for the quality, the consistency of their food. Of course, they care for safety. They like variety in their food products. What else? Nutrition and health, absolutely. Great. So we analyze food for nutritional components to be able to give health claims, to make sure that the food is the quality that the consumer is looking for, and all of that is good. Why do we analyze food from an industry perspective? We started talking about that last time. We gave one example, but do you remember? Can you say your name first? Jason. Cost, of course. So the industry is always looking to reduce cost and enhance profit. So they will be looking at replacement ingredients for that. Go ahead. Of course. So if we're changing an ingredient, it's changing formulation, how would that impact the product? What else they care for, Ash?
ensuring consistency between.
Consistent. Absolutely, consistency between productions. Sarah, compliance with government regulations. Absolutely, very good. Requires a clap for that. Government regulations is always important to follow guidelines, whether it is for the constituents or even the methods of analysis are regulated by government. And we'll talk more about that as we go. Okay, so this is all good. So this is kind of for you to not only to bring you back to what we talked about the lecture before, but also when you're studying, look at these recap questions and see, do I know this? Can I give examples about the particular question? Okay, so last time we gave example about product development. In this example, new processes. Again, in this case, they look for lower cost, they look for efficiency, speed. So they sometimes change equipment or replace old equipment. So this is an example which comes from real life. Actually, I was on sabbatic 1 year and worked at Cargill for a year, and this situation came to us. So they're producing soy flour for to be used as a medium for microbial growth, for nutrient, as a source of nutrient, for fermentation. And so the way that you have to produce the soy, you have to do heat treatment, because you want to inhibit anti-nutrients like trypsin inhibitor, for example. So there is some heat treatment that gets involved. So they replace the cooker. They brought in a new cooker, and this new cooker can control time and temperature better. It's more efficient, but it might change certain parameters within the soy flour. And true, the soy flour that was sold to a customer that were going to be using it for fermentation found that somehow the microbes did not grow as efficiently as they used to. So they came back to Cargill and said, something is off with your soy flour. So what food analysis test will be involved to ensure the consistency of such product when you change a cooker in this example? Lisa, shelf life. They're very good. That's a good question. So shelf life, basically, because the heat treatment impacts microbial growth that could compete with the microbes that we need to grow during fermentation. Yes, that is good. What else from a chemical composition? Brand, right?
Bread.
Yeah.
I don't know if it's related to it being a new cooker, but maybe like it's calibrated wrong, or like the temperature was like too high than anticipated, something like that.
Absolutely. You're thinking from an engineering perspective, from a process. Once you know the problem, then you go and solve that. That's good. But from a chemical perspective, that's assuring. Yeah. Moisture. So did the moisture content change because there is a heat temperature time that might impact moisture? But if we think of nutrients and we think of microbes needing nutrients to grow, what components would be important that might be impacted by heat? You had your hand up. What were you going to say? Say your name first.
Tam. So I was just gonna say nutrient content like got destroyed in general,
Yeah.
but
Can you give an example of one important nutrient?
yeah, but since they say soy flour is rich in protein, it could get denatured.
Yes. Yes, thank you very much. Yes, protein denaturation is a big, that was the main issue actually. So the main issue was the protein got overheated, it denatured, it polymerized, it was hard to digest by the microbes. So they had to go back and look at the engineering aspect of things, look at the temperature, the time, how can they control it, to bring back the parameters the way that the previous soy flour was produced. So for this, when I was working at Cargill during that year, that was what I suggested. Well, let's bring batches from previous soy flour from the previous cooker, and then a couple of trials from the new cooker to determine consistency between one trial and another. And let's look at protein denaturation from these different set of samples. So yeah, that was a real life example. Product complaints. Okay, this happens very often, especially when it's business to business. So you have the ingredient company and then you have the CPG company, and then you get the CPG company complaining to the ingredient company. So in this particular example, there is a cereal-based pallet that is being sold to a CPG company that takes this pallet and converts it into a snack food, specifically fried. So I added this in there, you didn't have it in your slide, but just to guide you a little better, that there is a frying in oil that happens there. And then the customer fries the pallet, convert it into snack food, package it, and sell it. Okay, so the finished product was rancid. So that customer went back to the ingredient company and said, well, there's something wrong with your ingredient. Our product is rancid. So you work for the ingredient company and you want to investigate the cause of the problem. So what do you do? Are you going to believe them right away that the problem is from your palate, or are you going to do some investigation? Or both? Ash.
I guess one thing that I would investigate is the like fry oil these, make sure that's being changed regularly. And that it's being used appropriately. For example, if it's not being put on
Yes, very good. Have you taken food chemistry already? And do you know all of this? Very good. Yes. So one big issue is you don't always want to say, oh, it's our fault. It might be your fault, but you want to look at other things that's happening. Oil is a big part of the processing of the snack food. So you would want to request a sample of the oil that was used to prepare that particular batch. Yes, they might have been cooking or frying in it above its smoke point, and smoke point, we'll talk about it later during fat characterization. Every oil has different smoke points, and if it's used multiple times, the smoke point will go lower each time because we are breaking the fats or the triglycerides into fatty acid, then the smoke point will gradually become lower, and then you would have oxidative rancidity happening. So that's one thing. What else would you think about that state your name?
Carol, you could
Carol.
do a packaging validity test, so if the quantity to fill.
Guys, you're very smart. Yes. So you will request packaged samples, and if you have, you will see if there's these packages are permeable to oxygen, and if they are, then of course there is some sort of fat in your sample. You will have oxidative rancidity. What else? Go ahead, Kevin. Yeah.
So I feel my first instinct also to like possibly not do any test is just you're selling it to multiple consumers and to see how your other consumers, if your other consumers are frying it as well, just ask them if they've seen any issue, because that'd be an easy way to not even, before you even test the company's oil,
Yeah.
you would just know if anyone else would have the same issue.
Absolutely. That is a very logistic thing that you can do. But the question is asking about analysis.
Yeah, I know. I'm trying to avoid
But
the type of analysis.
you don't want to avoid in this
Well,
class
because-
analysis now.
I think the reason also is if other people have it, then it's quicker for you to say it's our fault.
Yeah,
Versus if no one else has
Yes.
it, then you can ask for oil, and then you want, it's a finer way to look at it.
yeah. Do you have a job offer already?
No.
I think you will get a job offer very quickly with that mentality. Yes, definitely, that is true. But we're simplifying things here just to get the tests in this case. But yeah, definitely, if other customers have an issue, then do your palette. And for your palette, you want to go ahead and do rancidity measurements in your palette too. So you would look at hydrolytic rancidity, you would look at oxidative rancidity measurements, and we will learn all of this in a later chapter. Okay, good. Food forensics. I think, Kevin, you'll be really good in food forensic aspects. This is another example where a customer came in again to an ingredient company and said, there is a sharp matter in your product. So And in this case, it's true, there is a sharp matter in your, in your ingredient. And how would you investigate what that sharp matter is? Go ahead.
I mean, you could implement more metal detectors along the line to test that.
Yeah.
For like invest in X-ray technology as well.
Thank you. So if it is a metal object, you can run it through metal detector. If it is not a metal object, let's say you're suspecting something similar to glass or plastic, you would do microscopy or X-ray. Usually it's SEM, which is scanning electron microscopy, and then you would determine what it is. And in this case in particular, it was crystallized sugar. So it wasn't anything sharp. Yeah, so that is also a real life example, and it turned out to be crystallized sugar. Okay. Compete. That's going to happen quite a bit when you are in industry, whether you are producing an ingredient or you're producing a product, you are in most cases competing with other companies. And let's say you are Target, and you know how you go to Target and you find the same product, but it's Marketplace or something, whatever that is. And it's very similar to a General Mills cereal product, let's say, or any other company's product. So what is your first approach if you want to develop something similar to a competitor? The very first thing you would do, Lisa.
Would you do a sensory, like kind of analyze the product and like the texture and like physical characterization?
Physical characterization is great, Lisa, but before even that, go ahead, Priyashi.
Lower cost and reverse engineering layer one.
Reverse engineering, that's the key word. That's what one of the things you're going to be doing in lab next week. So you will first look at the package, look at the ingredients, look at the nutrition label, use a program to do reverse engineering. And then you can kind of identify what the formulation is. But obviously you want to achieve the same sensory and physical characteristics. So once you produce something similar, you run sensory acceptability and sensory difference testing between a competitor and your product. When you're doing a capstone project, some of you might be doing it this semester, some of you next semester. Sometimes you have to develop a product and then you run sensory tests, but you have to compare it to something. And then you will be comparing it to something already in the market. Okay, so government and regulations. So the government cares for certain things and regulates certain things. So one important aspect that is regulated by the government is nutrition labeling. So you see the nutrition label, it has certain components. Next chapter is going to be on nutrition labeling. So the government regulates what goes on the label and what tests are used to generate the information on the label. And there are good manufacturing practice and hazard analysis. So from a food analysis perspective, you want to make sure that the environment is clean and safe and sanitized for the production of food. And you also want to monitor hazard points, so to ensure the safety of the food product. So there is the microbiological analysis, but also there is the foreign matter analysis that we will learn in this class. There is also the chemicals of concern, and there is also something we call extraneous matter. So extraneous matter, in other words, filth, like a feather from a bird or a hair from a rodent. So all of these are regulated, and we'll learn about them in separate chapters, about 2 chapters later on in the semester. Also, you've probably heard of Code of Federal Regulations. So Code of Federal Regulation is a government-produced document or regulations that have information about nutrition labeling, also have information on standard of identity. So different food products have standard of identity, or different ingredients have standard of identity. They are all present in the Code of Federal Regulations. There is also codex, which is also kind of similar to Code of Federal Regulations, but really it has to do with international export and import. So any specifications for the products that allow importing and exporting are stated and regulated by codex. Okay. So analysis happens across the entire chain of events, starting from acquiring raw material or we call them just your initial ingredients, and then through processing, and as well analysis on the final product. So for the raw ingredients, basically, sometimes CPG companies ask for a certificate of analysis, for example. With every ingredient, there comes a certificate of analysis that tells you certain specification, moisture content, fat content, microbial load. Depending on the type of the ingredient, there will be information in the certificate of analysis. So the CPG company, instead of doing its own analysis, it relies on its select suppliers. So they would have like interaction with specific suppliers, and they trust them, and they trust their certificate of analysis. So they receive the ingredients, the raw material, and they don't do further testing. Sometimes they receive the raw ingredients and they have the certificate of analysis, they have the specification. Sometimes they do in-house testing, especially if they receive milk, they want to make sure that the milk is pasteurized. So they do, let's say, alkaline phosphatase test, which is an enzyme that can be inhibited during the pasteurization. If the enzyme is not inhibited, then they say, well, that milk is not really pasteurized. So sometimes they do certain tests. Oftentimes they rely on their suppliers, the specification and the certificate of analysis. So somebody has done the testing, either the ingredient supplier or the CPG company doing testing in-house. And then there are processing steps, and sometimes during processing you want to pull out samples and do testing. So I'm preparing yogurt, so I need to measure acidity or pH during the production, or I am producing flour samples, so and I'm removing the bran, so I'm preparing a white flour sample. So I will check the ash content, which would be an indication if the flour is completely refined or partially refined or still a whole flour. So I would do certain check. Sometimes I have a process control samples that I would test alongside the samples that I would pull online to do certain measurements. So lastly, you would do certain measurements on the final product. Measurements to ensure consistent quality. Maybe you do a quick sensory test, for example. Measurements that you have to do to comply with the nutrition labeling regulations and legal requirements. Reduced fat milk, is it really 2% fat? You want to make sure of that. Is it skim milk, 0 fat? You want to make sure of that. Acceptable to the consumer, with sensory tests. And then health claims. Are you claiming high protein? Are you claiming high fiber? Are you claiming a good source of calcium for osteoporosis? So you really want to make sure that your health claim is valid. Sometimes you say it's a rich source of, or a good source of, or an excellent source of. Whatever language you use, it corresponds to a certain amount of that particular ingredient. This was really important for students in the past years that developed projects, because the semester-long project had to deal with identifying tests on raw material, identifying tests during processing, and identifying tests on final product. And it was different based on the product that was selected. So as a food analyst, what do you really need to do or to know? So this is what we're going to study the entire semester because we're going to be treating you as a food analyst, and potentially you would need to know this information in your next job. So first of all, you want to know, what am I going to analyze? What do I need to analyze? So it's based on the situation. Am I needing to produce a nutrition label? Am I doing a food forensic? Am I addressing a consumer, customer concern? So I first identify what needs to be tested. Then you will learn in this class, for every analysis, there are multiple methods. So you will learn different methods for fat analysis, different methods for protein, for moisture, and then different methods to determine lipid oxidation. So there's so many different methods to select from for each type of analysis. So you will learn different methods, and as a food analyst, you will determine which method to choose based on different characteristics. And then once you identify the analysis and the method, you want to select a representative sample and you want to prepare it for analysis. There's a whole chapter on that, chapter 5, which is sample selection and preparation. We will learn about what does it mean to have a representative sample. So let's say you have 100 bags of flour, and each bag is 40 pounds of flour, and you want to test moisture content. Are you going to test the moisture content of 40 pounds, 140 pounds of flour? No, you're going to take a representative sample. So we're going to talk about how to take a representative sample so that when we determine the moisture content, we are certain that all of the flour in these bags have an average of 12% moisture or something like that. And there are certain steps to make sure that the sample is homogeneous, ready for analysis, and also stored adequately so that we don't change the characteristic of the sample until the analysis has been performed. Then we're going to talk about in chapter 4, well, no, I'm ahead of myself, analytical techniques. So we are going to learn chromatography, we're going to learn spectroscopy, we're going to learn spectrometry, we're going to learn atomic absorption, we're going to learn so many different analytical techniques that allows us to run the analysis and produce the data. And then in chapter 4, we're going to talk about data analysis and interpretation. All of you are required to take a statistics class, undergraduate statistics class. Here, we don't do a lot of statistics in this class, but we mainly get an average, a standard deviation, a coefficient of variation, and we analyze the data with only these simple measures. Then you make a decision. If you're working in industry, you collect the data, then you make a decision. The decision is, you tell the customer it's not our fault, it's your frying oil. Or you are testing a product online and then you find it damaged, you reject the whole lot and throw it away and start over. So you have to make a decision, and sometimes decisions are really important, and any mistake will cost the company a lot of dollars. So decision making is really key in this whole process. And again, no matter what, no matter where you are in the stage, there are government regulations that you have to comply with. Okay. So from what I covered so far, type of analysis to be carried out is determined based type of product, the health claim, the purpose, none, all, A and C. Just say your name.
I'm sorry, Hallie. All of it is relevant.
Yes, you think correctly. So it is all of the above. We want to know what type of product we have. Is it high in fat? Do I need to look at lipid oxidation? fat nut is not a problem, is the type of product has protein that I need to think about for fermentation, for example, like the example I gave you. Health claim, what am I claiming in terms of health? Purpose, is it a storage study? Is it nutrition label? Is it quality control? What is that? And that's how I determine the type of analysis. So all of the above. This again, recap question to help you study for this particular topic. All right, now I want to choose a method. And I want to make sure that method is valid or I can trust the results of that method. So I need to think about the characteristics of the method in different perspectives. So first of all, I look at the inherent properties of the method. We're going to spend some time and for chapter 4 to talk about specificity, precision, and accuracy. But let's see what you guys know about that. I know you heard the term precision and accuracy before, maybe specificity. So let me see what you remember. When I say a test is specific test, what does it mean? What does specificity mean to you? Any idea? Guess. No good guesses? You seem like you're scared. You have an idea, but yes, go ahead, Ash.
It's a method that would isolate one specific process or basically it tests one thing without.
Yes. Without other things.
I don't know how to describe
Yes, you are on the right track. You can learn, and what we call it, let's say, when we call it carbohydrate, there will be an assay that measures total carbohydrates. So most of the carbohydrates that you have in the system. And then you might need to determine glucose from total carbohydrate. You don't want to divide, you want glucose. So you would determine which method you want to use that would only tell you how many glucose you have. Not fructose, not sucrose, not starch, just glucose. So depending on the purpose of your method and what you want to know, you can determine which method to use, a highly specific method or a general method. So again, depending on your need. So in one of the labs, you're going to run a method that gives you total carbohydrate in a juice product or probably 7-Up and beer, actually, that was. So in 7-Up, you will determine total carbohydrate. So you have sucrose in 7-Up and you just want to know how much sugar you have. You don't have any other components that you care for, so you don't have to necessarily do an assay specific to sucrose. Total carbohydrate assay is sufficient because there's not very many other carbohydrate sources in that drink. So you can choose a less specific method if you know that your food matrix is simple, not complex. So you make a choice accordingly. Precision. What does precision mean to you? Go ahead, Haley.
Well, I think I remember in experimental nutrition, we like went between precision and accuracy and define them. And accuracy is like in comparison to like an accepted value,
Yes.
whereas the precision is comparison to like every other result that you can.
Absolutely. Precision is how close your data points are to each other. So when you do replication, when you do replicate, so if you're running an experiment, we always try to run at least 3 replications. And if your precision, say we measure it by standard deviation or coefficient of variation, if your coefficient of variation is like 20%, that's very high. So you don't have really good precision. It might be the fault of the method, is not a very precise method. There's so many steps and so many rooms for error. Or you need to learn techniques better and enhance your own experimental precision. Accuracy accuracy is you have if let's say you're gonna get assigned a product for proximate analysis section of this of the lab. And what we say, let's say you're doing cheddar cheese and you are measuring protein, fat, and moisture and ash. And then we do have USDA website, which you will be accustomed to in lab next week. You will see what cheddar cheese fat content is and what did you get in the lab, and you're going to calculate percent productive error, and then you're going to determine the accuracy. Sometimes the accuracy is not your fault. The method is not very good, doesn't give you good results. And sometimes you have poor accuracy because your technique is not that great. You're just starting to learn a method. You need to practice a little bit before you enhance your precision and accuracy. So it's not always the method's fault. Sometimes you need practice. All right, applicability of the method. So sample size. So sometimes a specific assay require a very small amount of sample. Sometimes it requires large amount of sample. So how much sample you need, and if we can apply this method. Reagents. Do you have the reagents in the lab that you need? Are the reagents expensive? Think about cost. Then if the method requires expensive reagents, then we look for another method. Do you have the equipment? So let's say you want to run mineral analysis. Do I have atomic absorption, or do I have ICP, which is another method or instrument that we will talk about later? So what equipment do you have in your lab? What's the cost related to running this assay in terms of reagents? Then you have a choice between different methods. Of course, time. Is it method that, okay, I'm measuring the efficiency of refining my flour. So I really need to measure ash content. Do I have an IR instrument that I can just put in my sample and get the ash content pretty quick? Or do I have to ash overnight, put a crucible, put the sample in, put it in the oven, come 24 hours later to get results? So what equipment I have, how much time I can spare, how reliable is this method? And do we really need this analysis, or we can run without this analysis? So you have to consider all of this before making a choice of the method selection. And all of this is summarized in your chapter 1, table 5. One last thing, I guess, I forgot about is personnel. So whenever you have a new method, you want to make sure that the people that are going to run the analysis are trained and they know the safety requirement, they know the procedure, you have good standard operating procedures they can follow, and you have the necessary safety precautions to take. And then objectives of the method. Okay, do I want to do nutrition label? If I need to do analysis to produce a label, I really need to use official methods of analysis. So official methods of analysis, meaning they have been tested, verified by an official agency, like the AOAC. It used to be called the Association of Analytical Chemists. It got changed to Association of Agricultural Chemists, but it still has the same AOAC. Not AOAC. AOAC is American Oil Chemist Society. AOCS. Let me write this down. AOAC is Association of Analytical Chemists, and now this became Agricultural Chemists. And then you have AOCS, American Oil Chemist Society. They're very similar in letters, so I get confused. So these two are examples of official agencies that verify methods of analysis, and once they're verified, you can use them for nutrition labeling, for example. And we call them primary, definitive, or reference. All of this, you can find these terms used interchangeably. They are called primary because they are often used to calibrate equipment. So let's say I have NIR that I use to get fat, protein, ash, and moisture. In order for the equipment to spit out these values for me very quickly, I need to calibrate it using official methods of analysis. Okay, so I'll give you later examples on other official methods of analysis than these two agencies. Sometimes it's just a rapid quality control. So I produced my, let's say, cheese, and I just want to confirm moisture content and fat content. I can do NIR or mid-IR on my cheese right away and get samples. We call these secondary methods. So we have primary methods, we have secondary methods. Secondary methods are calibrated by time, and they're usually fast. So this is similar to if I'm online and I need to measure moisture content, acid content, protein content while I'm producing. That also rely on fast methods, and we call them secondary methods that are calibrated by primary method. One example is that actually General Mills gave at one time when they are producing Cheerios, and you know, the sugar coated Cheerios, and then they have to monitor the crystallization of the sugar. They needed to have a certain amount of crystallization. So the official method is using differential scaling colorimetry. It takes time, and they needed something quick. So they started calibrating IR methods, infrared method, to give them quicker assessment of crystallization over time during production, and they call it secondary method of analysis. Okay. Sometimes your choice of method and the preparation is based on the constituents of your food product. Is the food high in carbohydrate? Is it high in fat? Is it high in moisture? And then there are certain steps and certain methods that you would follow based on the composition of the sample. There will be lots of examples during the semester, but here's one example. And I changed should to may. If you have your slides in your book, you change should to may. So we are determining fat content in a product that is high in carbohydrate and protein. They're not necessarily high in fat, but high in carbohydrate and protein, and you need to determine fat content. Do we need to do pre-drying? Do we need acid hydrolysis? Do we need solvent extraction, or may or may not need? You don't necessarily know this, but can you deduce an answer? Go ahead.
I would like maybe see the answer when you try to analyze that, so you can probably use solvent to extract that.
Yes. So C is one of the answers. Go ahead, Vish. Huh?
B and C.
B and C. Yes, and I'll explain. Sometimes also A. I'll explain why. So it is all of the above in this case. So why sometimes I need to pre-dry? You're going to learn that there are going to be two main official methods of analysis for fat. In the lab, you're going to learn a method called sock slit, and then you're going to learn another method called mojonnier in French, or moginer in how we twisted the French into English. So the sock slit method is a semi-continuous method, and there's no shaking. So in this method, if the moisture content is above 10%, if you have really high moisture content in your sample, the moisture would interfere with the solvent reaching the fat. So your sample needs to be pre-dried in that method. So later on, some of you might have cheese as a product assigned. Cheese is high in moisture. All cheese samples will be pre-dried before analysis with the SOX. But if we use Mojo, short form Mojo name, if we use Mojo, you don't need to pre-dry because there are steps that ask you to shake, shake, shake. And then the solvent will reach the fat and you don't need to pre-dry. Now, in this particular example, I said high in carbohydrate and protein. So if you have a matrix that is complex, you have high carbohydrate, high protein, fat will be entrapped in this matrix. So if you hydrolyze by acid, you can break down the carbohydrate and protein and release the fat for easier solvent extraction. So here's a table that I pulled from somewhere. I think from your book at some point, before, not the recent edition, I think edition 4. So what you see here, dried eggs, high in protein. When we do acid hydrolysis, you get 42% fat. Without acid hydrolysis, you get 36% fat, so you underestimate fat by 6%. Flour, you also get a little higher fat content, but anyway, the fat is low in flour, and it's 1.73 compared to 1.2, but since it is a very low percentage, 1.73 percentage-wise is higher than 1.2 by at least 30%. So when you have high carbohydrate or high protein, you need to break out the matrix so that you can release the fat, so you can do your solvent extrusion. Yes.
Just to clarify, so like, and so I get it all together. So no matter what, though, you're still doing a solvent extraction. It's just what you do before that.
Yes, but not, well, we will learn that some assays for fat are not solvent based. There are other assays. So that's why actually I changed the should to may in this question to avoid confusion. But many of the fat analysis method requires solvent, but you will learn about a Babcock method that is for dairy product, does not require solvent. Validity of the method. So we look at validating the method by looking at how specific, depending on our need, how precise, how accurate, the variability and repeatability of the result. Let's say I ran the assay today, I'm going to run it next week. Am I going to still get the same or similar results? So this is very important. If the method changes every time or gives you different results, then it's not valid, not reliable. Let's say you're developing a new method, and this happens a lot in industry and in academia. We always develop new methods for speed, so efficiency, safety, cost. But when you develop a new method, you have to validate it. You have to run multiple samples, look at accuracy, look at precision, look at specificity, and then compare it to an approved method or to the old method. So you have to always validate your new method. And sometimes in companies, they utilize services such as standard reference material. So they can obtain standard reference material from the National Institute of Standards and Technology, NIST. So that institute, the National Institute of Standard and Technology, produces standard material, whatever it is, depending on your product or ingredient. They send you a sample. You run the your new method, and then compare your results to the results that they give you using the previous methods. Or you can run the old method and the new method side by side on this standard reference and see how close your results are. Sometimes you can send it to sample services, or we call it check sample service. So, AACCI, which is now known as Cereals and Grains, which used to be American Association of Cereal Chemists, International with an I, now it's called Cereal and Grain. They are also an organization that provide check sample services. AOCS is another organization that provides such service. What do? They provide you with, or you get your standard reference, you run the analysis, you send the analysis to them, and they check the data and do the analysis for you and tell you whether or not your method is valid. So they provide you that service. Sometimes you don't do any of that, you just have your own internal control samples and test an old method and a new method with your control samples to validate and ensure that this new method will work. So here, oh, I went over a minute, but I'm gonna finish. Go ahead, Ash.
I didn't want to interrupt you, I just wanted to let you know you're.
Oh, thank you. But yeah, I did look at the time, but here, nothing to worry about other than showing you that there are official methods of analysis produced by different organizations where you can find official methods. This is specifically for cereals. This is for oils and oil products, oilseed products. This is for milk or dairy products. And that's it. Thank you. Thank you.
You're, I don't know if you did or what's these yet, but those are all just last year's.
Oh,
I wasn't worried, I just didn't know if you saw it yet,
no, so anything old, don't worry about. We will
Yeah.
update them
as they go, okay.
yeah,
Perfect.
yeah. Because when I
I
asked,
already had so many messy assignments.
missed assignment
Yeah.
already?
That's what I was like, no way.
No. Yeah, thank you for letting me know.
Okay.
Take care.